Immune cells. Types of immune cells: phagocytes, lymphocytes, and other cells. These include memory and plasma cells. Phagocytes: neutrophils, macrophages, and phagocytosis. Phagocytes. These cells are produced and stored in the bone marrow through a person's life. They are distributed around the body in blood. They act as scavengers, removing dead cells and invasive microorganisms. Macrophages make up 5% of white blood cells in the body. Neutrophils make about 60% of white blood cells in the body. Phagocytosis is the process of destruction of microbes by phagocytes. Macrophages. They are usually found around and inside organs, removing foreign material. They travel in the blood as monocytes, which later develop into macrophages. Once they leave the blood through the gaps in the wall of the capillary. They are also responsible for initiating immune response. They don't destroy microorganisms. Instead, they cut them up and display their antigens. This process is called antigen representation. Neutrophils. They travel throughout the body and often leave the blood by squeezing between fine capillaries too. They move through tissue, engulfing pathogens, and during infection are released in large numbers. Phagocytosis. Cells under attack release chemicals called histamines. These chemicals mix with the waste released by pathogens to form chemicals attracting neutrophils. This process is called chemotaxis. Neutrophils destroy pathogens by phagocytosis. This includes moving towards the pathogen clustered and covered by antibodies. They recognize the antibodies as they have receptor proteins on their cell surface membrane. They attach to the pathogens. The cell surface membrane of the neutrophils engulfs the pathogen. It engulfs and traps them within a phagocytic vacuole by the process of endocytosis. Lysosomes fuse with these phagocytic vacuoles, releasing hydrolytic enzymes, breaking down the pathogens. After digesting the pathogens, the cells die. These dead neutrophils collect at infection sites, forming pus. This is the process of phagocytosis. Lymphocytes. B and T lymphocytes. These are smaller than phagocytes and easily recognizable by their large nuclei that fill their cells. They are produced before birth and stored inside the bone marrow. Only mature ones can carry out immunity. They are specific and specialized to respond to only one type of antigen. When mature, they circle the blood and lymph. This ensures distribution. They come in contact with phagocytes and one another to give an effective immune response. T cells coordinate immune response by stimulating B cells to divide. These B cells secrete antibodies in the blood. Antibodies recognize antigens on pathogens. This helps to destroy them. Other T cells seek and kill any infected cells by contact. B lymphocytes. They remain in the bone marrow until maturity. When mature, they spread throughout the body and become concentrated in lymph nodes and the spleen. Each gains the ability to make one type of antibody molecule specific to one antigen. Each one divides to create a clone, which is a small group of identical B cells. They use a part of the antibody molecule to make a cell surface membrane receptor. They combine with the specific antigens, forming antibody-antigen complex. Only a small number of B cells with the cell surface membrane receptors complementary to the antigen are stimulated and divide by mitosis. This is clonal selection. This small group of specific cells divide repeatedly by mitosis. This is called clonal expansion. Huge numbers of identical B cells are produced over the first few weeks following primary infection. Some of these B cells become plasma cells and secrete antibodies into the blood, lymph, and linings of the gut and lungs. They are very short-lived and die after a few weeks. The antibodies secreted also remain in the blood for short periods of time. Some other B cells become memory cells, which remain circulating in the blood for long periods of time. This means if the same pathogen is reintroduced, these memory cells rapidly divide by mitosis, developing into plasma cells and even more memory cells. This is repeated on every subsequent invasion caused by the same pathogen, carrying the same antigen. This inhibits pathogens before symptoms of the disease occur. This is the process of B cells. Immune responses. The first immune response is slow, and few B cells are specific. This causes fewer antibodies to be produced. The second immune response is rapid, and large numbers of B cells are specific. so more antibodies are produced. This diagram shows a clear representation of the number of antibodies in the blood. Antibodies are globular glycoproteins with a quaternary structure, forming a group of plasma proteins called immunoglobulins. Each antibody consists of 4 polypeptide chains, 2 heavy and 2 light chains. A disulfide bond holds the chains together. Each antibody has 2 identical variable regions. The sequence of amino acids in these regions makes specific 3D shapes binding to a specific antigen. A hinge region allows the flexibility of an antibody molecule. This allows it to easily bind to a pathogen's antigen. Antibodies act as labels to identify antigens. This allows phagocytes to destroy them as they become marked. Some antibodies are called antitoxins. Their function is binding and blocking released toxins to prevent any damage. The functions of antibodies. They combine with virus to prevent damage. They attach to the flagella, inhibiting the movement and coating bacteria. They punch holes in cell walls, causing darker pressure to burst the cell. They also cause agglutination of bacteria to prevent spreading. They also combine with toxins, neutralizing their effect, antitoxins. T lymphocytes. They have specific cell surface receptors with a similar shape to antibodies. They are also specific. Unlike B cells, these receptors, however, are only activated if they recognize the antigen on one of the host cell, the infected individual. This occurs due to antigen representation of macrophages. Complementary receptors on T cells respond by dividing by mitosis. They also go through clonal selection and clonal expansion. T helper cells. When activated, they release cytokines. These cytokines stimulate appropriate B cells to divide, developing into plasma cells and secreting antibodies. Cytokines also stimulate macrophages to carry out phagocytosis more violently. They also stimulate T killer cells to divide by mitosis, differentiating by the production of toxic vacuoles. T killer cells. These cells search the body for antigen representations, attaching to the cell surface membrane of these cells which are infected, secreting toxic substances from their vacuoles, killing these infected cells. This also kills the pathogens inside. This is the process of the division of T cells by mitosis, and its maturing in the thymus and blood. Other cells, memory cells. They are produced and remain in the body. Antigen representation on host cell causes the stimulation of T cells. T killer cells divide by mitosis, punching holes in the membrane of infected cells, secreting toxins such as hydrogen peroxide, killing the cell and pathogen inside. T helper cells secrete cytokines, stimulating appropriate cells to divide by mitosis, forming plasma and memory cells. Both T killer and T helper cells produce T memory cells. Thanks for watching. Do you have any questions? Leave them in the comments. Don't forget to share with friends, and to subscribe would be very much appreciated. This allows you to be notified whenever a new video drops.